All right, thank you so much, guys. I appreciate it. So today we're gonna do digestive system 2. We're pretty much gonna look at the latter half of the digestive system, and so we'll look at what you looked at the stomach last time. That was kind of like the last organ we looked at. So we'll look today at the small intestine. We'll look at how the secretions are regulated, and we'll look at the functions of the the small intestine as well. And then we'll look at most of the accessory organs like the liver. We'll talk about that and the bile and how it's secreted and so forth. I know in the lab we've already started talking about anatomy of the liver, and that will come in handy when we talk about how bile is conducted throughout the system and how it ends up in the duodenum. And then we'll talk about the large intestine, which is really split into like 4 parts: the cecum, the ascending colon, transverse colon, descending colon, and in the human we got the sigmoid colon, all right, and then the rectum, and then the anal canal, and then we'll just talk about the functions of the large intestine and so forth. So really, the major organ of the digestive system would be the small intestine. It's very long, okay, 21 feet long. Thereabouts, give or take. Okay, so small intestine, the major digestive system organ. All right, and it really completes all the digestion. In the stomach, we talked about amylase and some of those enzymes and peptidases that break down carbohydrates to disaccharides, right? Maybe fats to mono or diglycerides, right? And then also the protein is broken down to smaller chains called peptides. So the small intestine will finish it up, break the disaccharides into monosaccharides, and we'll talk about those enzymes, and we'll talk about the regulation of those secretions and so forth, from the pancreas, from the small intestine itself, and then from the liver. All right. So transfer of calories, feed, right? We got the duodenum, and we know about that, jejunum, ileum, all right. And then we get the colon, the large intestine, really, which is split into the cecum, which is the very first part, and then the ascending and descending and transverse and sigmoid colon in human. The cat doesn't have sigmoid colon, so in the cat we'll just be doing ascending, transverse, and okay. To the ileocecal valve, the ileocecal valve, the part where the ileum gets into the large intestine. Like I was saying, we got a little bit about 21 feet long, really, with the ileum being the longest. Okay, 12 feet, and the jejunum about 8 feet. Okay, this is about 1 foot long, about 25 centimeters. So, and the duodenum cradles the head of the pancreas. The pancreas is split into the head, the body, and the tail. And the tail end of the pancreas is next to which organ? Anybody want to say the organ that the tail of the pancreas is next to? No, spleen. Yeah. Remember, the spleen is always on the side like that, looking like a cone. Yeah, so that's where it ends, because the pancreas is very long. Remember, when we talk about in the lab, we say pancreas, and we go all the way across. It's a large organ, because it's got 2 parts actually, an exocrine and an endocrine part, and we'll talk about that. So just pretty much know the basic anatomy of that, and today we'll talk about the functions mostly. What is this organ right here? Yeah, the gallbladder. And then, of course, large intestine, and so forth. And this is the small intestine, of course, with the mesentery and so forth. And we'll talk about that. I like these pictures because, you know what, they show cadavers, real cadavers. So you get a chance to see that. That's why I always put them in there, so you can look at them on your own, and really get to see. So we'll kind of talk about these already today. The book calls these, you see, this is what we're calling the bile duct, left and right bile ducts, okay? Which is also correct, but the book calls it left hepatic and right hepatic, okay? And then the one that joins up is then the common hepatic. Okay, so I just wanted to talk that in advance so that you don't get confused when you get there. Okay, so the common hepatic is what we're calling the bile duct. Okay, all right. The rest of the stuff is the same. It's just that part. So this we're calling the cystic, the one that comes from the gallbladder. We're calling the cystic duct because this is going to come in handy when we talk about the conduction of bile, because I'm going to need you to know about that. The conduction of bile from the gallbladder, where it's stored, all the way to the duodenum. All right, and we'll talk about it when we talk about the liver, and then the common hepatic duct or the bile duct and the cystic duct join to form the common bile duct, and that is the same as we've been doing. Okay, and then this is the duodenum, you can see, and it's cradling the head of the pancreas. That's why when I was saying if I put a pin next to the small intestine that's close to the pancreas, you know it's duodenum. Okay, the shortest, and this will cradle the head of the. And then of course there is the ileum right at the end. They just cut it short so that and there is the deal. So this is the cecum right there. The cecum and the vermiform appendix, which is this, they're both blind-ended tubes. Okay, that start off, and their function is really to counter the bacteria that colonizes our gut, right? And they do quite a bit of things. The digestive system, in the digestive system, those bugs, we talk about it when we mention the vitamin D. So they really make vitamin D for us, vitamin K, those bugs, okay? And they digest the cellulose that we cannot digest. And so it's a mutually beneficial, beneficial symbiotic relationship between the two of us, okay? But then there's other things that the bugs do, which we'll talk about, that we kind of don't like, but they got more benefits than no benefits. So the small intestine's job is to absorb and to finish the digestion. So one of the biggest things about it is to increase surface area. Okay, we all know about the microvilli. Said, oh, the microvilli and the villus and so forth. So the way it can upregulate, or increase the surface area 600 times, 600-fold. That's astounding. So first we have this sort of circular, motions. Oh, I had to spit out. I was reading. So these are the circular folds that kind of increase surface area already. Okay, instead of just a straight line like that. You see how this line is going to be much shorter than these two when you pull it together. So that's the number one. The way you get to 600 times. You know what I'm saying? Okay. Then you have villus on it. See what I'm saying? On those circular folds, you have villus on it. Okay? You got villus on there. You're not done yet. Okay? And then you got microvilli now on that. Can you imagine that? This is how you ratchet the area up to 600-fold. Do you see that? So, microvilli, and then villi, and then circular folds. Okay? So you come up to here, 600-fold. Okay. Sometimes I might ask you, how does the small intestine increase its area up to 600 times? Then you talk about that, and that could be a 2-point question with a bonus freely for 3. Right? Okay. So microvilli are the tiniest, and so it really ratchets the area all the way up. So we're able to explain that, and of course. The epithelium, which is the cells lining the small intestine, which is called the mucosa, right? And underneath that, you learned the same layers when you did the stomach, right? The submucosa is underneath there, and then there's all these lymph nodes and whatnot. Look at those, what we call pia stretches and nodules, and then the muscular layer. Okay, you have the circular muscles and the longitudinal muscles that go along lengthwise. And then you have the adventitial, the connective tissue covering on the outside. So same 4 layers just go throughout. Okay? And so it's columnar epithelium. Those are, those cells. And those cells also form the absorptive cells. which we'll talk about in a little bit. We'll talk about the cells that are pretty much lining. So you will have like an absorptive cell that will go like that. It's columnar, right? And then it will have a nucleus, and then it will have the villi and so forth, right? And microvilli. Okay, so it's columnar epithelium, those are the cells, okay? And then between the villi, it's got these crypts or these sort of indentations where you have intestinal glands in there, or pits, little pits, okay? So we'll talk about the cells that are involved with the epithelium, in a little bit. There's 4 cell types, depending on their function. And their function is more intuitive that you won't get lost and it makes sense too. All right? So, and this is the villi. You see, this is very magnified, quite a bit. And on those villi would be the microvilli, which you need much more higher magnification to see, you know what I'm saying? And so there you go, microvilli on the villi. And it will ratchet it up. So you can see the absorptive cell, and then it will have all that. So the absorptive cells will be lining the villus like that. Okay, and then this will be then the microvilli. You see what I'm saying? You see the villus will be like so. And then you have a columnar absorptive cell like that. You see what I'm saying? And then you've got that. Then this will be the microvilli. You see what I'm saying? Isn't it amazing how the body kind of is already arranged? It's totally amazing. Okay, so this is the histology I was talking about. We've already talked about the absorptive cells that are lining there. And then, of course, doing it will be those cells like the goblet cells that produce the mucus. All right? So you get the absorptive cells with the microvilli, right? And then wedged in between them in the gastric pits and so forth, you've got some of these other cells that produce the mucus or the hormones and so forth, okay? So goblet cells produce mucus. They do the same in the stomach, right? You saw that before, so it's nothing really new here. And then the granular cells, sometimes called Paneth cells, these are like the macrophages of the small intestine, okay? Macrophages of the small intestine, because you need to protect from bacteria. Any system that takes from the outside and internalizes is a risk of infection, so it's got to be guarded with all these immune cells, okay? And later on, we'll learn about specific immune cells in the liver and so forth and so forth. All right? So I want you to remember these cells. Palate cells, protect from bacteria. So these are the macrophages of the small intestine. And then, of course, the endocrine cells, you know what the endocrine cells do. They produce hormones, right? They produce regulatory hormones. There's a bunch of hormones that the small intestine will secrete, like secretin, cholecystokinin, all right? Intestinal lipase, all those things. Okay? So, those cells do that. So, gotta know the histology of the small intestine, right? All these cells and their functions. And their functions is absorptive cell, you think of the macrovilli, right? Because the absorption, you need to increase surface area, right? So, if you just think of the structure of this, you know what the function of it is. And the name kind of tells you what it is. And you know that you need to have a microvilli to increase the surface area for absorption. So goblet cells, you kind of know they produce mucus. Why would you need mucus up in the small intestine? Why would you need, what's the main function of this mucus? What would it do? What is, judging from the secretions from the stomach.
Acid?
Yeah, acid. Because when the chyme enters the small intestine, the pH is like 1. So that's the most acidic you could get. Right? Almost, 0 is like very acidic. But 1, so that's very high acid right there because of the hydrochloric acid that's released by the stomach. So you need protection immediately because by the time you secrete some of these hormones that will cause bicarbonate ions to come from the pancreas, bicarbonate ions to come from the gallbladder, because the gallbladder also releases bicarbonate ions. You kind of need protection right away. So that's the big function of that, so that it doesn't corrode through the. So when you have ulcers and things like that, you're having problems with these goblet cells. See what I'm saying? Because they're not there, and when you get the acidic stuff, it will start to eat up your digestive system. Okay? Even in the stomach, same thing. You have a problem there. So, and know the functions and these cells, sometimes they ask about that. And then the Peyer's patches, if you like, because this is the guy who talked about it quite a bit. And so these are aggregates of lymphoid tissue. We kind of talked about it, lymph nodules, okay? And they're packed with all these T and B cells, you know, to protect, okay, on top of the having granular cells, which are the macrophages, specifically of the small intestine, okay? So, gotta know that, you know, this histology and all this structures in there. And this, I like to just show this. You can kind of look at it when you have time. You can see the mucosa with the villi and then the submucosa and so forth. Submucosa, that's where you have those nodules, those lymph nodules. The Peyer's patches are located in the submucosa, which is a bit of connective tissue and so forth. And then, of course, the muscularis, the muscular layer, which is, of course, got, you know, the longitudinal and circular muscles shown here, all right? And the longitudinal muscles shorten. The circular muscles do what are called segmental contraction. I mean, and we'll show that to you in a little bit, okay? And the longitudinal and circular both do the peristaltic movements, and I'll show you how, you know, which are, you know, move material along the GIT. You can see again the villas and the and this green part of the lacteals, right? Remember the lacteals for fat absorption, which we did, I think, in one of the examples. We talked about that, the lacteals. Yeah, because we talked about the lymphatic system, right? Because the lymphatic system, one of the functions, apart from draining fluids in the extremities, it involved in fat absorption. Okay, so they'll kind of show up again now when we talk about how you process fat. After the lipases take care of it, and then it goes into the lymphatic system. All right, so that's that. So mucus, of course, protects against the digestive enzymes as well. Because they're protein, right? They digest protein, and those cells, those absorptive cells, have got proteins on their membrane. Don't get digested. You know what I'm saying? So you need that too, apart from protecting initially from the acid of the stomach before you neutralize it. Okay, that's why the enzymes are released in what kind of form? From whichever organ makes it. The enzymes are released in what kind of form? How are they released? Are they released active or non-active? You know, the enzymes. Inactive. Remember when you're doing the secretions of the stomach, pepsinogen and stuff like that? Okay, pepsinogen will be the inactive part. If you release it active, it's going to start digesting away. It won't stop. You know what I'm saying? So you need to release it in an inactive form. Then you activate it.
It becomes pepsin.
Yeah, then it becomes pepsin, then it will do the job. When it's inside, when there's a lot of these are amino acids, and of course, the thing is already protected by the mucus lining of the small intestine. Then you become active, okay? That way they will only digest the food, not yourself. Because that would suck really, you know, if you digest through your own intestines and then, you know. So, like I said, the small intestine will finish it up. All those, you know, the triglycerides, some of them remain, some of them get broken down to biglycerides and monoglycerides, and those will be finished up. All the lipases, intestinal lipase will break down anything. If it's a triglyceride, it will be broken down to glycerol and 3 fatty acids. If it's a bi, you know, 2 broken down to glycerol and 2 fatty acids. It's gonna be finished here, okay? And all, of course, all the, you know, disaccharides, sucrose, fructose, glucose, maltose, glucose, all those were broken down to the most simple parts. And all those peptides, the short protein chains that were broken down by peptidases in the stomach, broken down to what? Amino acids, okay? Which is the simplest form. And so that's why the intestine is major, because it will not only finish the digestion, but what else will it do? What else does the small intestine do, guys? Absorb. Yeah, absorb. So these are 2 major functions. If I was to ask about that in the exam, right, you won't forget, right? Absorption 2. So it's got major functions there. So what we're doing here, most of the mechanical digestion occurred where? Most of the mechanical digestion, where did it occur? Huh? Mechanical digestion?
The
What is
mouth.
mechanical digestion? Yeah, in the mouth. I'm checking to see if you cannot remember what you did before, because it's important. Mechanical digestion starts in the mouth, and a bit of chemical digestion through which enzyme?
Amylase.
Yeah, salivary amylase. Okay? Yeah. So we get things that will stimulate intestinal secretions, which will be the chemicals that are contained in there. The chyme, okay? One of the chemicals, the big one, is the HCl, that will cause secretin to be secreted, which will cause bicarbonate ions from that pancreas and from the duodenum. I mean, from the the gallbladder. Okay, and then of course tactile irritation as well. It's like touching. Remember tactile? We talked about those touch receptors, like the Meissner corpuscles and so forth. They must be lining the small intestine, right? Because touch, when something touches something else, when you physically put something in there, it's gonna cause the tactile stimulus. So, what kind of receptor would this be? The tactile one. Yeah, touch, but it's grouped under what? The umbrella of it. Chemical is because of chemicals, right? When you do touch, it's because of mechanical. Yes, thank you. Mechanical. So that would be a bunch of mechanical receptors in there, so for touch and so forth. Okay, and then of course a chemical, because there's a bunch of chemicals coming in there, and that will stimulate secretions. Okay, and then the vagus nerve always does that. Vagus nerve is cranial nerve number 10. Yeah, the vagus. Guys, you can't forget that. Those cranial nerves, don't do that. Okay, so the first hormone, of course, that the small intestine is going to secrete based on those stimuli, the chemical stimuli, and of course, the tactile. The chemical being the major, of course, the chemical to detect acid. So secretin will then go and cause the pancreas to produce bicarbonate ions. All right, and I'll show you how some of these bicarbonate ions are basic, so they will neutralize the acid. All right, they'll neutralize the acid. All right, so just know about how the small intestine is stimulated, to make the secretions and to start acting. All right, and then of course, for it to be successful, it's got to do what? It's got to have mixing movements for start, to mix the chyme with digestive enzymes and so forth. Okay, so then and I'll show you how the mixing movements, the segmental contractions, mix the material. And then of course you need peristaltic contractions to move material along, right? Because this is all 21 foot of it, it's gotta go. All right, so if I ask about the kind of intestinal motility and what kind of functions that they do, to say segmental will mix, right? You gotta mix the stuff up first, okay? Then peristaltic will move material along, okay? In fact, when you have a bowel movement, it's a major peristaltic movement in your colon. You know what I'm saying? And we talk about at the end about how that's stimulated. Mostly after you've eaten a meal and you put new stuff in, you gotta take old stuff out, so it comes up. All right, so we talk about that at the end.
Lovely chapter.
It is. It talks about all the good stuff and the bacteria too, and I'll talk about what the bacteria will do. Okay, so contractions, of course, are stimulated, just physical distension. When you dump material in a thing that's elastic like that, it will start to distend. And then, of course, it will know something must be there. And then, of course, so remember, we talked about the enteric nervous system when we were talking about the organization of the nervous system. There's a lot of nervous and processing here, and it's all automatic. And all these are reflexes that stem from the lumbar enlargement of the column, of the spinal cord. Okay, so there are reflexes that are local reflexes because of all these things going on, and because of how it has to act quickly without needing the central nervous system control. So it really has its own little nervous system down there. Because if it doesn't act quickly, it's going to be digesting itself all over the place. So you see, there's actually a branch called the enteric just for that. So for all these local reflexes that happen in there, when it distends, when it's got chemicals in there, you start to release immediately. Okay? And then, of course, from the central nervous system, we get two kinds of stimulation. The parasympathetic, which in this case will increase digestive function. Okay? Because this is what you do in the resting state, in a resting state. Okay? When you're not running away from a lion and stuff. And then the sympathetic will tend to kind of sort of stimulate other things during flight, and kind of sort of down-regulate digestive activity. Okay? So this is the only time when you kind of have to do a backflip, right? About that. Parasympathetic is going to be stimulating this stuff. Okay, so don't forget that. Keep that in mind. When we start to talk about these reflexes, I want you to remember that. Because the tendency is that parasympathetic inhibits, not for this one. So, and then so distension of the cecum. Remember, the cecum is right at the junction of the ileum and the colon. So, distension of the cecum will cause closure of the ileocecal sphincter. See what I'm saying? Is it making sense? When you get the cecum to distend, why would you then close the ileocecal valve? Think about it. It's kind of counterintuitive.
You're waiting for it to all back up and then it'll open and down.
Well, this material is coming into, it's not coming out. It's coming into. When material comes into the cecum from the small intestine, from the ileum, right, at the ileocecal junction.
Oh,
right?
so it closes up so it doesn't
do
one-way valve.
Yeah, it's a one-way valve.
Yeah.
So that, yeah, but why would it close when it starts to distend? Yeah, one of the things is that it's a one-way valve for sure, because you don't want material to back up. Okay, so why does it close when it starts to fill up, when it's distending? Okay, why?
It's like, I don't know, so you can build pressure.
So it will regulate entry of material from the small intestine. You don't want to just dump a whole bunch of 21-inch long, 21-foot long stuff material. It's going to flood it. So, because then it's final processing that's happening. So it's like a matter of aliquoting it. It's a matter of doing a little bit at a time. When it is distended, it is filled, then you have to close it. You see what I'm saying? You have to close it. So that you don't keep dumping material in there. That's when you might have a diarrhea situation if you, because diarrhea is to flush. So if material before the colon, in fact, that's how diarrhea occurs. When the colon doesn't have enough time to absorb the fluid from the chyme, then it's going to be flushed out. You know what I'm saying? So especially when you have a viral infection and stuff like that, that's when it happens. But normally, it will close up so that it will give time to process the material that's just been dumped in there. Okay, so that's why I asked about it, because it's kind of counterintuitive about this regulation of the ileocecal sphincter. Okay, distension of the cecum will actually close it normally. Okay, so these are some of the things that I could sort of ask about in the exam in terms of how you read your reasoning behind some of these actions. Okay, all right, very good. So this is what I was calling the segmental, contractions that will do what now? Mix the material along. So the circular muscles, if it will contract there, and then it will contract there. So what's going to happen when it contracts there? Material is going to move this way, material is going to move that way, and then material is going to move that way, material is going to move that way. You see, when you squish it like that, it will do that. So these segmental contractions will go all over the place. And the material will keep going like this. So it keeps getting swished like this, swished, and that's how it mixes. You can see how this dark brown thing is, talk about brown when we're over there. Brown. Okay, and then you see how it's fading, it's getting, because the segmental contractions will occur anyway, and it will move in material this way, that way, this way, that way, and it goes here, it goes there, it goes there, it goes there. And before you know it, it's all mixed up nicely. So that's how it will mix with the digestive enzymes. So if I ask about this process of segmental contractions, you can talk about that, how it will contract with the circular muscles, because then those are the ones that crimp the small intestine, okay? And then those digestive segmental contractions will occur randomly along the length of the that small intestine, and then therefore it will mix, because material is going to squish to the left and to the right, laterally, whenever a segmental contraction occurs. And if they are occurring randomly throughout, then you're going to have all this, and then it's going to mix. Okay. So now we go to the liver now. Remember, we already talked about how the liver has got the left and right lobes, median lobes, and then the quadrate and the caudate lobe. Okay, and then internally it's divided into 8 segments, right? And the liver cells are hexagonal. Okay, hexagonal. It's got 1, 2, 3, 4, 5, 6 sides. Okay, and then it's going to be divided into lobules. Those 8 segments of the liver are then divided. You got the 4 lobes, and then you got the 8 segments of the liver. And then each liver is divided into lobules, okay, which are the functional unit of the liver. Okay, the functional unit of the liver, it will have like a branch of the bile duct, you know, coming from the left and right hepatic or left and right bile ducts, and so forth, from the left and right side. And then it will have a branch on each corner, on each part of the hexagon. And it will have the branch of the aorta carrying oxygenated blood, all right? And then it will have, called the hepatic triad, the branch of the portal vein. The portal vein is carrying what? Material from the small intestine, taking it to the liver. Because material from the small intestine, you've just absorbed all sorts of stuff, and you don't want to distribute into the system immediately. You want to take it to the liver so it gets processed, where it will do a lot of things. And we talk about just now about the functions of the liver, where it will process nutrients, change them into other nutrients. It will detoxify, you know, and so forth. So, so that's the portal triad. So on each of those 6 corners, you have the portal triad, with the this carrying the bile from the you know, the liver sinusoids, and you know, and so forth, and the bile canaliculi, which join up to form the sinusoid. To form the branch here of the bile duct, which then carries it to the duodenum. Okay, via those things we talked about in the anatomy part. Okay, those ducts. Okay, and then this will be coming from the, you know, from the intestines, carrying all those absorbed, and it's oxygen poor but nutrient rich. Okay, in the branch of the hepatic portal vein. Okay, so the liver lobules are a structural and functional unit of the liver, and each of those has got that hepatic triad. All right.
What are the 3 parts of the portal triad again?
The 3 parts will have the branch of the aorta carrying oxygenated blood to the liver cells, the hepatocytes, and then it will have the branch of the you know, the bile ducts, the bile canaliculi will join up to form, you know, so that's why that's the green one with the bile, because green bile is kind of green. It's a yellow-green kind of fluid, and then the branch of the, so the bile is going away from the liver because the bile is produced in the liver. All right, and then the hepatic portal vein will branch into that blue dot, which is the branch of the hepatic portal vein, carrying from the small intestine nutrient-rich foods towards. So this is towards the liver, this is away from the liver, this is towards the liver, because it's carrying oxygenated blood to the liver cells, right? And then this one will be carrying deoxygenated blood, but full of nutrients, nutrient-rich. So those are some of the things I'm going to want you to talk about when we talk about conduction of bile from the liver to the intestine, and then conduction of blood from the small intestine carrying all those nutrients but oxygen poor to the liver to get processed. Okay, and I'll show you a nice diagram here in a little bit. But note the functional unit of that is the lobule. So we got the 4 lobes, then you got the 8 segments. 8 segments are split into these lobules, and the lobules is the functional unit, and it's hexagonal, and it's got those portal triad on each corner. Okay, so it's composed of these hepatocytes, liver cells, you know, with plates radiating outside. Okay, so you have the hexagonal plates like that, and then, you know, kind of going like that. And then in the middle will be the central vein. Okay, the central vein. So this will be stacked, and then you let me draw this as blue actually, because it's going to join the central vein, and then it's going to go to the hepatic vein. The hepatic vein now is going towards the heart now. Then the hepatic vein joins the inferior vena cava. Okay, but that's after the blood has been processed and dealt with in the liver. So that's why we have to, that's why we need the portal system. What did you say a portal vein is? A portal vein is, it's gonna do what now? Did we say that? We did say that.
Yes. But when you take the deoxygenated
Yeah.
blood to the liver, but it's got the nutrition.
Yeah, so it's leaving one set of organs, right, which is namely the small intestine. So it's got the capillaries in the small intestine. So it takes blood from the capillaries of the small intestine and takes it to the capillaries of the liver. So that's a portal. That's a portal blood vessel right there. Because it will drain one set of capillaries and drain into another set of capillaries. Okay? So that's what that is. Alright? So, but then you have the hepatocytes here in the middle. Hepatocytes, and columns. And these plates radiate outwards like that. And the hepatocytes will be lined. So it will be a column of hepatocytes like that. Okay? Radiating outwards from the central vein. Okay, in that fashion. And at the end, you have a portal triad, carrying fluid to and from, namely oxygenated blood and deoxygenated blood with nutrients, and also bile away from the liver. So that's the portal triad. So if you were to describe a lobule, a liver lobule, I might say, okay, describe the functional unit of the liver. So then you talk about the portal triads. Talk about how it's hexagonal, and it's got columns of hepatocytes, all right, that are stacked up, divided by these columns, okay, radiating from the center, like that. Okay, but really, I would like you to talk about the portal triad and how you have the hepatocytes that just sort of arranged in this hexagonal way, and then talk about what the portal triad is comprised of. Okay, then that would be good for me already. Okay, if I ask about the functional unit. So I mean, this will. I like to show this because in the lab we see the cat liver. Okay, so this is the right lobe where the gallbladder is, the left lobe, and you flip it on the other side, and this is the inferior vena cava kind of goes in through there, and then the falciform ligament right there, and then you get the quadrant lobe over there, and then the quadrant lobe right here. Okay, so those are the four. Okay, so you can kind of look at it later. So there you have the liver right there. So this is what I was talking about earlier on. Okay, when I first started, this is what we were calling the left bile duct and the right bile duct, the left hepatic and the left and the right hepatic ducts, which will join to form the common hepatic duct or the bile duct. You can say both. I don't really mind. Okay, I'm just it is the same thing. Hepatic, of course, is referring to the liver, and bile is, of course, referring to the bile, because the bile is what is being conducted. Okay, left and right bile duct, left and right hepatic duct, same thing. Common hepatic duct, same as the bile duct, that we're calling the bile duct. So in the lab, you can call it either way. I don't mind. But the rest of the things is the same. The cystic duct comes from the gallbladder. So the common hepatic duct or the bile duct and the cystic duct unite to form the common bile duct. The common bile duct is one that goes to the here they kind of forgot the common one. Common bile duct goes to the duodenal ampulla, the papilla, the duodenal papilla, where the duct, the major duodenal papilla is where the duct of the pancreas also opens in there to the same part. But the pancreas has other accessory ducts that open into the minor duodenal papillas. Okay, and I'll show you that. But I just this part of the anatomy you kind of know about it already because we've been doing it in the lab. And then it's just that you haven't seen the location of the caudate and the quadrant lobes. Okay, in the human, because we get the cat all the time. Okay, so you can kind of look at that later just to sort of compare and contrast the cat's liver and the human. Okay, I wish you could see kind of aspect. It's kind of expensive, but one of these days we might be able to visit the lab. But the portal triad, we already know about it. It comprises of the hepatic duct, all right, conduct bile towards the duodenum, small intestine, right? Because remember the head of the pancreas is cradled by the duodenum, right there, where the duct of the pancreatic duct opens, so does the hepatic duct, or the common bile duct, right? So the hepatic duct will be a branch of the, of that the bile duct. Okay, so hepatic artery, of course, branch of the aorta, coming in with oxygenated blood from the heart, because the aorta goes into the descending aorta, thoracic, abdominal aorta, and from there you have branches. You have the splenic artery, you have the hepatic artery, and then the hepatic artery will then branch up and distribute blood in the liver, right? Okay, and the portal, hepatic portal vein, the vein that is just drained all the small intestine and descending towards the liver will have branches. Those are the branches that you find on the portal triad. Those three little dots are blue, the blue, the green, and the red one. Okay, so that the hepatic portal will then branch up and do that. So carries blood from the visceral, the small intestine here, and takes it to the liver to be processed. And then when we talk about the functions of the liver, it will all make sense. Why the blood has to be conducted to the liver first before you distribute it broadly throughout the systemic circulation. Okay. So the hepatic cords are those columns of those hepatocytes that radiate from the central, vein in the middle of that lobule. All right. Remember, I drew those columns of tissues. So it's like a column of tissue that radiate, and it's made up of the hepatocytes. And then the hepatocytes, of course, in there will be the bile canaliculi, which then join to form the branch of the hepatic, the bile ducts that go to the small intestine, right? Because the bile is made by those hepatocytes. So those hepatocytes we have this canaliculi that sort of go through collecting the bile and then sending it to the end of the portal triad, to that branch of the bile duct. Okay? Which will be the hepatic duct. Okay? Alright, so, and then the liver has got this sort of capillaries, very wide capillaries with big holes of them, so that blood can be filtered through. And those sinusoids are specialized capillaries that join to form to join the central vein. So those sinusoids again will kind of be radiating like this, or over, I thought I was picking blue, but it's okay. And then they join in the central vein. This will be the hexagonal part, whatever. Okay, so they all radiate like that, and the bile canaliculi also radiate going out like that, and then to the portal triad. Okay, and then everything from the branch of the hepatic portal vein will be conducted towards the central vein. Okay, I should have drawn this. Okay, so this will be the sinusoids like that. Sorry, and then this will be the central vein in that lobule, right, in the middle. And then we get also in there because we just absorbed all this blood from the small intestine, right? The phagocytes of the liver, which are called Kupffer cells. Okay, what are the phagocytes of the small intestine? Anybody remember the phagocytes of the small intestine? Pannate cells or granular cells. Okay, you see each of these organs that has all this stuff on the outside. It's got its own mark. And then soon when we do the lungs, we'll talk about the dust cells. Okay, which kind of makes sense too because of the respiration like that. So Kupffer cells are the hepatic macrophages that will be getting these sinusoids, which are literally like capillaries well, not capillaries, but the small veins, or venules, if you will. But these are big, and they've got huge holes on them, so they're called sinusoids. So the light space is full, like with the epithelium that and the phagocytes, Kupffer cells. So know the Kupffer cells, which are the phagocytes of the. Okay, so this is what I was talking about. You see the columns of the hepatic cords. You see the hepatic cord here, and then you got here the branch of the hepatic portal vein, and then it's joined to the sinusoid. You see the sinusoids. Okay, the branch of the hepatic portal vein and the branch of the hepatic artery join up. Okay, the blood will mix, and then it will all go to. And then of course, the oxygen in there, in the red one, will be used by the cells as they do their job. And then, but then it will be venous blood anyway, and it will be going back to the heart via the central vein, which then joins from the hepatic vein, which then goes to this vena cava, right, inferior vena cava. Okay, you see what I'm saying? And then the bile canaliculi are draining bile that's being made by all these hepatocytes. You see this hexagonal stuff? And it's going to the end, to the portal triad again, going towards the duodenum now. Okay? It will all be collected by the left and right hepatic or left and right bile duct, which joins to the bile duct or the common hepatic duct. Okay? Then it goes into cystic for storage in the bile, in the gallbladder. And then whenever you eat a fatty meal or whatever it is, or anything actually, because you need bile secretions too, then the bile will be caused to contract, usually by cholecystokinin, and then it will release the fluid into the cystic, then goes into the common bile duct, which then goes into the duodenum. And I'll show you pictures, and then you can visualize it, okay, in a little bit. But I want you to kind of sort of see these lobules. Okay, they got the hepatic cords, which are columns of hepatocytes, which themselves are hexagonal. And then lined in there are bile canaliculi that will be draining the bile that's being made by the hepatocytes and goes to the end, to the portal triad, where it becomes the hepatic duct, okay, which then goes into the left and right ducts, hepatic ducts or bile ducts, and then the common hepatic duct and the cystic, okay? So I'll show you a nice diagram that shows this, okay? But I want you to be able to have a good appreciation of the lobule, okay, and the structures that make up the lobule and what the functions of it is, okay? Especially the portal triad, because the portal triad is the one that's distributing blood, okay, to the liver, oxygenated blood and deoxygenated blood with nutrients, and also the hepatic ducts with the bile canaliculi are draining the liver of the bile, okay, so that it goes into storage in the gallbladder. Okay, so that's the part I really want you to understand, okay, the lobule, the functional unit. Because if you get that, then the rest of it will kind of come with the lab, like the cystic and the hepatic ducts and the bile ducts, you know that. Okay, so what does the liver do? It produces bile. Okay, the gallbladder just stores it. Okay, just like the urine is made up in the kidneys, and then the urinary bladder just stores it. In fact, I need to go to the bathroom, but I had to come to class, so I'm gonna wait, and we'll talk about how it's regulated earlier in terms of the reflexes. So what does it do? It stores and processes nutrients. What does the liver do? It will turn all that absorbed glucose from the small intestine into glycogen. Okay? So that's when the pancreas will produce insulin after you've eaten a meal. So it will make sure the hepatocytes take in that insulin, and then they will process it into glycogen for storage. So that's storing nutrients. And you might eat nutrients in certain ratios. Okay, maybe you've eaten a protein that has a certain kinds of amino acids, and maybe you need a different kind of amino acid. So what's the liver going to do? It's going to change that one amino acid into the proportions that the body will need. Because there's no way you're going to take in exactly the kind of amino acids you need, exactly the kind of fats, and exactly the kind, you know what I'm saying? you're gonna take in what you can, you know, and the food will have the building blocks, and then some of those building blocks are edited. And one of the big ones is conversion of one amino acid by changing the R group. All you have to do is change the R group on an amino acid, right? And then it's another amino acid. Okay, so the liver will do that so that those nutrients are in the proportions you want. Okay, so that makes sense. It can also synthesize new molecules. Okay, the liver will do that, synthesize new molecules. All right, just like we just talked about in terms of changing one amino acid into another. You know, and if you are on the Atkins diet and you're taking a whole bunch of protein, does it mean you don't need carbohydrates and fats? You do. So the liver will make sure all those proteins will be processed, will be cycled to make carbohydrates and lipids. So that's synthesis of new molecules. Okay, in the exam, if I ask about the liver functions, I want you to kind of talk a little bit about that. Don't just say produces new molecules and stop right there. Tell me about how it will convert proteins into carbohydrates and lipids and so forth, or how it will convert certain amino acids into other amino acids. Okay, and detoxification is really simple. Sometimes you eat harmful chemicals. You know, you need to detoxify those so that you don't get toxic things going on in there. Okay, so that's and we already kind of talked about this when we talked about blood. We said the macrophages in the liver, mainly those Kupffer cells, will do what? Will break down worn-out red blood cells. We already did that, so we already know that function of the liver. So it's nothing new. So some of it you already know about. If I was to ever ask about the functions of the liver, you talk about that, and then of course they will get rid of the bacteria, and then other debris, cellular debris too. Okay, it's digested by there, it's broken up like that. So functions of the liver, there's plenty, and you should be able to explain them and kind of talk a little bit, flesh it up a little bit. And then production of blood components, such as, what are some of the blood components since we just did blood? Blood proteins, albumin, produced by the fibrinogen. What is fibrinogen involved in?
Chlorine.
Chlorine. What is albumin involved in? It was in the exam, right? But it's the concentration, plus osmotic concentration of blood. Okay, it's mostly involved. And what do globulins do? What are globulins? Globulins? What about immunoglobulins? I just thought, right? Immunoglobulins, what's the function? It's immunity, right? So the globulins are those immune cells, like the antibodies and stuff. They are produced in the liver. Okay, blood components. So if you talk about producing of blood components, you talk about that. Okay? Don't just say the liver produces blood components and stop there. I'll just give you half a point. Okay? So you gotta talk about that, okay? And some of these functions are kind of intuitive. You've kind of seen them before already. It's now just kind of sort of putting it all together in one lump. So branches of the hepatic artery, of course, and the hepatic portal vein and the triads will kind of unite. And then remember, like I was saying, and then they form the sinusoids, and then the sinusoids will then go and join the central canal, right? So remember, from the branch of the artery and the hepatic portal vein branch, at the triad, they unite. So the deoxygenated blood and the oxygenated blood mix, and then the cells of the liver will process that, the nutrients, and then will take the oxygen. And then the sinusoids, remember, they go and connect to the central vein, right, which is in the center of that lobule. And then the central vein connects into the hepatic vein, right? And then the hepatic vein goes into the inferior ventricle. So that's what all this is really. Okay? Starting with the triad, where you have the branch of the artery, aorta, right? And then you have a branch of the hepatic portal vein. They join to form the sinusoids, hepatic sinusoids, and the hepatic sinusoids feed the central vein in the middle of the lobule. Central veins from different lobules unite to form the hepatic vein, right? Then the hepatic vein goes and connects to the inferior vena cava. Okay? So that one is not so, it's not so bad in terms of, and then the vena cava will jump into the heart. And then that's how you circulate it. So this is pretty much what it's talking about. It's not talking about anything new, but it's just showing you the, the bile coming from the bile canaliculi, which form the hepatic duct, right? The hepatic ducts from different lobules will then form the left and right hepatic ducts or left and right bile ducts. Common bile, or I mean the bile duct or the common hepatic duct, then the cystic, and then the common bile duct when it goes to the duodenum, right? So this is that way, okay? But we start off from the hepatic artery, which is a branch of the aorta, taking blood to the liver, right? Oxygenated blood, and then a branch of that hepatic artery and a branch of the hepatic portal vein are right there, the portal triad, and then they unite to form the sinusoids, right? And then the sinusoids take the blood to the central vein, then the central vein goes to the hepatic artery, hepatic vein, and then the hepatic vein to the inferior vena cava. That's it. So this way and that way. All right. So, all right. So, and you can kind of look at that later, but pretty much bile from the liver. So this is how it's conducted, right? So the bile canaliculi that line up those between the sinusoid, between the hepatocytes, okay, goes and forms the hepatic duct, which is the part of the triad, hepatic triad, okay? And then those hepatic ducts join up to form the left and right hepatic ducts, or left and right bile ducts, if you want, okay? Which then forms the bile duct or the common hepatic duct, all right? And then it goes to the cystic duct. The cystic duct comes from the gallbladder, where it is stored, right? And if the gallbladder contracts, then blood goes through, bile goes through the common bile duct to the duodenum, right? So that's what that is really. All of this is that, okay? From the bile canaliculi draining those lobules, and then the hepatic duct, right? Which will then take the bile, that's the green one, and then we will join up to form the left and right hepatic duct or left and right bile ducts, right? And then the common hepatic duct, which then goes to the cystic. Cystic goes to stop, and then the common bile duct will take it to the duodenum. And then it will open at the hepatopancreatic ampulla, all right? It's called the hepatopancreatic ampulla, and it opens into the major duodenal papilla, okay? It opens into the major duodenal papilla, but there's some other accessory ducts, smaller ducts from the pancreas that open into the minor, duodenal papilla, okay? But the hepatopancreatic ampulla is where the major pancreatic duct and the common bile duct open together into the duodenum, okay? So know these terms. The hepatopancreatic ampulla is that part where it enters, okay? And so this is the conduction of the bile again, from the left and right hepatic ducts or left and right bile ducts, and then goes into the common hepatic duct, all right? And then cystic duct here, and then goes into the common bile duct to the hepatic, hepatopancreatic ampulla, okay? And then this is the major duodenal papilla. And then you see some of the smaller ducts, accessory ducts of the pancreas open into the minor pancreatic ampulla, okay? So this is pretty much what we did in the lab, right? Pretty much in terms of conducting. And then you can kind of go through this again later on, okay? Same thing here, shows all those ducts again, and then it goes to the major duodenal papilla, and you can examine those pictures of cadavers later, okay? So now gallbladder and how, the gallbladder, of course, what it does is store the bile, okay? It's a small sac, inferior surface of the liver, all right? It stores and concentrates bile, okay? stores and concentrates bile. Okay, bile, of course, it's kind of like a greenish, yellow green. That is alkaline because of the bicarbonate ions. All right, and then it's got all these neutral fats in it, and then it's got phospholipids, cholesterol, bile salts, major component, bile salts for fat emulsification. So sometimes I ask about the components of the bile, and then of course electrolytes. Right, electrolytes are those charged particles, right? But neutral fats will be kind of like those triglycerides or whatnot, because they're neutral, they don't have a charge. Phospholipids are charged because they've got a hydrophilic head. Okay, so that's what neutral fats are, that's all it is. Okay, so know the components of bile, and then what is the bile salts do? They emulsify fat. Okay, you know how fat is kind of like lumped into these big droplets? It kind of sort of coalesces like that. So what does the bile salts do? They emulsify it into smaller fat droplets that can be almost soluble. You know what I'm saying? Yes.
When you have gallstones, does that mean that bile salts aren't working right?
When you've got gallstones, you've got a lot of cholesterol in there, because the bile salts are salts of cholesterol and some other charged particle on this, on some of these electrolytes. So that makes the salts. Okay, so yeah. So if you have a lot of cholesterol and you're kind of processing that, so you form these very solid stuff that look like even as big as a dime. They could be as big as a dime. Those stones are, a cousin of mine had them removed, and they're big stones like this, of calcium and all these ions with the cholesterol. Okay, so it happens if you've got high cholesterol. So usually those people, when they get problems with gallstones, they eat less fatty diet. Yeah, you need a good diet, you need a lean diet. Okay, so because if you're eating a lot of fatty diet, your liver will want to make more bile, and then it's got all this cholesterol in it, and then if you're not processing it correctly, then it forms those salts become like calcium carbonate, on those things. It's one of those things, and it will be the same thing but made of cholesterol and the charged ions. So it makes the gallstones. So if you eat less fat, then you don't need it to make more bile. So then it reduces the probability of you forming the gallstones. Right? So because it helps to make cholesterol soluble as well. See, whenever you sort of add a charged particle to a non-polar particle, it becomes soluble. You see what I'm saying? So that's the deal. So if you have a lot of cholesterol and you're going to try to make that soluble, then when it gets mixed with these charged particles, those electrolytes, to make it more soluble, then you form those stones. See what I'm saying? Sometimes it's not even that you have a fatty diet, it's genetic. Okay?
Is it?
Mm-hmm. Sometimes you may have problems with just processing your cholesterol. So, like this other comedian, Griffith, Eddie Griffith, he's a very lean guy. So, skinny, you wouldn't think you would have high cholesterol, but he had high cholesterol. It's just genetic. It could cause cardiovascular problems or things like that, or gallstones. And then it could block the ducts of the pancreas and all that, and then the pancreas can become inflamed. Then you could start to be diabetic because it's what's producing the insulin and it's being inflamed. So it can ratchet up into big problems just because of the stones. Okay?
But I don't know where to tell you that. I have a friend that has it really bad. She needs to get it removed, but they won't help her do that.
They should remove it.
Yeah.
It's not a big surgery. You just open it up like that and then you take them out.
I think her medical is an issue.
Excuse me. Yeah, that's another big thing about.
But they don't tell her that stuff either. I think it's genetic, and it could cause pancreatitis.
Pancreatitis is bad. Pancreatitis will bring a bunch of problems. Glucagon is made in the pancreas, insulin is made in the pancreas, and all those things.
So a person that has gallstones for a while could end up being diabetic eventually.
Yeah, you could end up with all sorts of slew of problems. Because if the ducts from the pancreas, because the bile is coming down those ducts and it's blocking that ampulla, it's blocking all those things, you can't secrete the enzymes fine into there. But the pancreas becomes getting inflamed, and it's not making insulin anymore, it's not making all these other things. Okay? We're going to run out of time today. Enterohepatic circulation makes sure the bile salts are recycled, because it's not going to be making this and just throwing them away. And over 90% of those salts are recycled, okay, via the enterohepatic circulation. So the bile salts absorbed in the small intestine again, go back into blood, go back into the duodenum. I mean, back into the liver, and then they kind of reprocessed and packaged back into the for storage in the gallbladder. Okay. So what happens is that when the bile goes into the because it's carrying the bilirubin, because the bilirubin is coming from what? From heme, right? From heme, from after the breakdown of hemoglobin, which is what we said the liver does. It's gonna carry that bilirubin, and then when it goes into the small intestine, that's when the bacteria in the small intestine will digest it into those bilirubin derivatives, of which one is urobilogen. Okay, and then that's what makes the brown color of feces and makes the yellow color of urine, because they're absorbed back. So we kind of know about that already. Okay. But for here, I want you to know the components of bile, and of course what the bile salts do, which is emulsification of fat. All right. And also making fat cholesterol more soluble, because cholesterol is, after all, what? A lipid, right? So it's not soluble. Okay, so regulation of bile secretion. So, like I was saying, the duodenum produces, the small intestine produces secretin, okay? And also cholecystokinin. So cholecystokinin will cause the gallbladder to contract and release bile salts with the salts. Cholecystokinin usually responds to the fatty kind. So if you eat like a McDonald's burger or whatever, you eat a lot of fat. McDonald's. I don't like McDonald's, by the way. I keep saying that because it's the most popular thing. But I don't eat McDonald's all the time. Okay, so it will cause because you get fat and you need emulsification of fat by the bile salts. So cholecystokinin will be released to secrete bile, mostly with bile salts, for emulsification of the fat. Okay, and then secretin is also released by the small intestine in response to the acid. And it will cause the gallbladder to release a watery solution with bicarbonate ions in it so that it will neutralize the acid. You see what I'm saying? You gotta do two things. First, you need the chemicals that are in there, what kind of food chemicals need to be processed. Okay, then you also need to neutralize the acid, always. Okay, from the small intestine. So, know the function of cholecystokinin, okay, which causes relaxation of the hepatopancreatic ampulla, because that's how the, the bile is going to get into the small intestine, and then of course, as a result, then bile will enter the duodenum. So bile salts and secretin, because they'll be absorbed. The bile salts, like I said, in the hepatopancreatic circulation, or the enterohepatic circulation, will be recycled and go back to the liver. When they go back to the liver, it's like a positive feedback mechanism, will cause more secretion of bile. You know what I'm saying? Kind of like a positive. Until all the fat is digested, then it all stops. Okay, so bile salts will also stimulate the gallbladder to contract. All right, and then of course vagus nerve, cranial nerve number 10, will also cause weak contractions of the gallbladder. Okay, all right. So that's how it's regulated by cholecystokinin and secretin. Okay, and also from the parasympathetic system. If I ever talk in the exam about how the bile secretion is regulated. So you want to talk the parasympathetic, you want to talk cholecystokinin and say that it causes bile salts, and then you want to talk about secretion, that it causes the watery alkaline with the bicarbonate ions for neutralizing the acid. And then when the bile salts are absorbed in here too, they go into the bloodstream, and then they come back to the liver, cause more secretion of bile. Okay, and that's how they get kind of recycled. Okay, so that's the. And then, of course, anatomy and histology of the pancreas, right? The pancreas has got two things. It's got an exocrine function, which produces the enzyme. Exocrine means it's got ducts, right? Remember when we talk about glands, exocrine have ducts. So, and that produces the enzymatic component, okay? The enzymatic component is secreted through the ducts, through the exocrine ducts, to get into the duodenum. Then it's got the endocrine function, the pancreas does. And then it's got what are called the islets of Langerhans, where you have the beta cells making the insulin, and I'll show you a nice diagram here. And then the alpha cells making the glucagon. Glucagon is what turns the glycogen into glucose. When you're hungry and you haven't eaten a meal, you secrete glucagon, and the glucagon will break the glycogen in the liver to form glucose. When you've eaten a meal, you secrete insulin so that the hepatocytes taking the glucose into blood, turn it into glycogen. You see what I'm saying? So there are these zymogen cells. So the acinar part, which is like the alveolar glands, they are kind of lobular, and I'll show you in a little bit. So exocrine function produces enzyme, and it goes into the duodenum. The endocrine function, endocrine means secrete within. Does endocrine have glands? Ducts? No. So that means it goes where? Endocrine will secrete into blood, inside, into the blood. Okay? Why is that? Why do we secrete in the endocrine part, like the insulin and the glucagon, go into the blood? Why doesn't the insulin and the glucagon go all with the enzymes into the duodenum? What is insulin? What kind of compound is it? What is glucagon? Insulin is a protein. That's why when you're taking insulin, if you're diabetic, what do you do with it? Do you take it per horse through the mouth? You inject it into yourself. Because if you take it, if you drink insulin, what's going to do? You're going to digest it into the amino acids. Because it's a protein. So that's why you have the endocrine function, which secretes into blood.
Some diabetics
They wouldn't contain insulin. They would be containing something else that helps with the insulin upregulation.
I see.
Yeah. Because if you take insulin, it's a protein, it's going to get digested. Then you won't have any insulin anymore. Okay? So I want you to realize that. That's why you have the two endocrine and exocrine functions. And we know about the endocrine and exocrine functions because we did glands, and we know that exocrine functions, they've got ducts, and the endocrine don't have ducts, so they secrete into blood. Okay? And that's because those glucagon and insulin are proteins. So insulin beta cells, glucagon alpha cells. Okay? On the islets. So, I mean, this is the pancreas with the head cradled by the duodenum, the body, and the tail. Okay, this is a CAT scan because the fat on a CAT scan is black. Okay, instead of an MRI. An MRI is the opposite. The fat will be white. Okay, but I won't go into that. But anyway, this is a CAT scan, so it's showing you the internal organs and so forth. And then you see the islets of Langerhans will have the alpha cells and the beta cells, and then it goes into the bloodstream. It doesn't go into the ducts. And the acinar part is what forms the enzymes. All right? You see what I'm saying? So the pancreatic secretions, again, the pancreas also from the small pancreatic ducts, you know, the small pancreatic ducts that join to form the major ducts will produce the bicarbonate ion-rich solution. Okay? So the secretin will cause those pancreatic ducts, small pancreatic ducts, to produce the bicarbonate ions. Watery solution with bicarbonate ions neutralizes acid and provides optimal environment for the pancreatic enzymes. Otherwise, enzymes will not work in an acidic environment. All right? And the enzymatic component, which is produced by the acinar cells, all right, which is the exocrine function of the liver, those are conducted into the duodenum via the duodenal papilla, major duodenal papilla. And they're called acinar because they look like lobes. Remember, alveolar and acinar glands look like that, and then you have the acinar cells like that, and then, of course, forming the enzymes, and then they go through the ducts, and then they end up into the major pancreatic duct, which empties into the duodenum. Right? So just know that the aqueous part, which contains bicarbonate ions, is made by the small pancreatic ducts, and then, of course, the enzymatic component is made by the acinar part, or the exocrine function of the pancreas, right? And then the endocrine function will produce the glucagon and insulin, which are proteins, so therefore they can't go into the digestive system, they will be digested. They go into the blood. Endo secrete within, secrete into blood. All right? Okay, secretin, of course, like we said, will cause that pancreas to produce bicarbonate ions. Cholecystokinin, on the other hand, will cause enzymatic components from the acinar cells. So cholecystokinin stimulates the acinar part of the pancreas. Cholecystokinin stimulates the bile salts component of the pancreas. Okay? Secretin does the same thing in the bile, the gallbladder, and in the pancreas in terms of producing the bicarbonate ions. Okay? To neutralize the acid. So know about these two enzymes, what they do in the gallbladder, what they do in the pancreas. So parasympathetic stimulation, like we said, for the digestive system, you have to forget what you have learned so far. It will increase digestive activity, parasympathetic. And then, of course, sympathetic will, some of them will stimulate the secretion of enzymes a little bit, but it's mostly the parasympathetic system that's more active here, okay? So, and then that's the cycle for the cholecystokinin and the secretin. So just know what the cholecystokinin and secretin is doing in terms of the what it's kind of secretions it's causing from the pancreas. You gotta be going because the other class that comes in here doesn't like it. So, okay, the large intestine, we may have to finish this later on, but the main function of the large intestine is to absorb the fluid, right? And then, so like I said, the large intestine is separated into the cecum, the ascending colon, the transverse colon, the descending, and the sigmoid colon, and the rectum. The rectum is what stores the feces before you have the defecation reflex, and then it will go, okay? And then the anal canal is the last part. The anal canal is guarded by the internal sphincter, which is opened reflex-wise, and the external sphincter is one you open voluntarily when you want to poop, right? Yeah. Okay. So anyway, that's the large intestine. The vermiform appendix is so called because it looks like a worm. Vermis is a worm, like the vermis of the cerebellum, okay? And then of course the ileum. And then you can look at this. I think we can finish this next time, real quick. The large intestine, because the large intestine has got three parts, like the tenicolli and the rostra that we don't see in the cat, and also what are called epiploic appendages. And then that's what we'll cover next time, okay? The epiploic appendages are sort of pouches of fat whose function is not well known, okay? So we'll kind of sort of pick up from this. I don't think there's many slides left after this. The large intestine really doesn't have much. So we can quickly brush through that next time. Okay, guys? Thank you so much. I apologize, but we'll finish next time.